now. Okay, well, I think bisphenol A doesn't need any introduction to this audience, but just to remember, the polycarbonate plastic is done by bisphenol A, and we know that these ester bonds are hydrolyzed in the presence of high temperature, basic or acidic media, and then the monomer is released, and the monomer is in many daily products in producing that bisphenol A can be found in the urine of 93% of US citizens, so practically everybody has bisphenol A in his body. And the concentration found of bisphenol A in human serum is this one here, which is in the nanomolar range, between 1 and 18 nanomolar. Bisphenol A can act in cells, binding to many different targets and doing different things depending on the concentration, but this classically we view bisphenol A as a weak estrogen. And a weak estrogen following this classic definition of estrogenicity. The concept of estrogenicity of an exogenous chemical is based on the property of these compounds to bind ER alpha and ER beta and to act subsequently as transcription factors when binding to the estrogen response element in the DNA. And indeed, this is how they work. So nuclear receptors are normally outside the nucleus, or in some type of cells in the nucleus, but outside are bind to heat shock proteins, and when the ligands bind to it, they dimerize, they go to the estrogen response element, they bind to it, and they incorporate a number of cofactors. And this is the situation at the end. And the transcriptional activity not only depends on the ligand binding to the binding site, it depends on the ligand, depends on the sequence of DNA, and depends on all these cofactors that are regulating the chromatin, the opening of the chromatin to activate transcription. And all that changes from cell to cell and changes as well depending on the metabolic state of the cells, for instance, and from many other signaling. Okay, so that's the point. But in any case, it was demonstrated years ago, and this is an example, that transfecting cell lines with ER alpha or ER beta, bisphenol A behave as a weak estrogen when transcription was measured. So we can see here that it's needed to go to 1 micromolar to start having a transcriptional effect. And indeed, in this same paper, for instance, it was demonstrated that the binding of bisphenol A to ER alpha and ER beta in this transfected cell was very low. And indeed, the bisphenol A has an affinity of 10,000-fold lower than that of estradiol for both ER alpha subtypes. And indeed, the estrogenic potency of bisphenol A in vitro is 1,000 to 5,000-fold lower than that of estradiol. So it is a weak estrogen. The problem, and these authors discussed this in their paper, is that this couldn't explain results such as the one that is here, that was from a paper in 97 already. This is the activity of BPA and estradiol at low concentrations, releasing prolactin in vivo. So they couldn't explain how BPA behaves as a strong estrogen in vivo rather than in vitro, and there are more examples about that. And the point, or I think one of the main points, is that this definition here is correct, but it is incomplete. So we know now that estrogens not only act by binding at ER alpha, going to the nucleus, acting as transcription factors, and binding to an ERE. The situation, the scenario is much more complex, and we know that by sure now, and I'm going to show some of the examples. So this is a paper that was published by the group of Cheryl Watson like 20 years ago. And they demonstrate that ER alpha at the membrane was located at the membrane and could bind estrogens when located at the membrane. Already in 1998, we demonstrate that estrogen plasma membrane receptors were able to signal from the membrane and to produce physiological effects, such as the release of insulin at physiological concentration of estradiol, 100 picomolar, 1 nanomolar. And indeed, this is a paper from 2000. We demonstrated, I think for the first time, the rapid response of a xenoestrogen, in this case bisphenol A, 1 nanomolar, acting on the intracellular calcium signaling in mice islet of Langerhans. So these are calcium oscillations, is the ones that you see here. This is an islet of Langerhans, and we are measuring calcium in the whole islets. Blue is low calcium, and yellow-red is high calcium. So what BPA does very rapidly, in a matter of minutes, is to increase the frequency of the calcium oscillation. Each time that there is a calcium wave here, there is a release of insulin. So the higher the frequency of intracellular calcium spikes, the higher the amount of insulin that is released, and we will go back to that. So BPA at low concentration can produce very rapid effects. And now I'm going to pass to this cartoon that is from Ellis Levin, an Ellis Levin review. Ellis Levin has done an outstanding amount of work in the field in the last 15 years, and he has really demonstrated how molecularly ER alpha can work outside the nucleus. And this is the model that he proposes, and is the palmitoylation of a part, like 5% of the ER alpha, which is expressed in a cell, is palmitoylated. It goes to the membrane, and then it forms a signalsome together with other proteins, including SARC, G proteins, etc., to signal through the plasma membrane. And this is very well now known in the field of estrogen and accepted by the whole scientific community. So the scenario that we have for estrogens is not the classical view, and we published that already like 13 years ago. So is this. These are the nuclear classical effects that estrogens binding to ER alpha, ER beta, dimerizing, and acting as transcription factors. But we now know that ER alpha and ER beta are acting outside in the cytosol, also associated to the membrane to activate other pathways, kinases, G proteins, increasing intracellular messengers, and that is going to activate other transcription factor, and it's going to be able to regulate transcription, but starting outside the nucleus. Okay, and when they are outside the nucleus, as Ellis Levin showed before, all what is surrounding the receptor is different. Very likely the ligand binding site is going to behave different as well. And in addition to that, there are other membrane receptors that can bind estrogens and now have been molecular characterized as GPR30 or GPER. Okay, so we have used a cellular model for many years to demonstrate how ER alpha and ER beta can bind BPA and can produce some alterations in the pancreatic beta cell. The pancreatic beta cell has two main functions. On the one hand, to produce the transcription of insulin gene and therefore to biosynthesize insulin, and on the other hand, to release insulin in response to extracellular glucose. Well, now I'm going to concentrate in the first of this function, which is the regulation of insulin biosynthesis. So already in 2006, we demonstrate that injecting 100 micrograms per kilo per day during 4 days to male mice of bisphenol A or estradiol, the insulin content of the islet of Langerhans increases. This is vehicle-treated mice, and these are estradiol or bisphenol A-treated mice. So these islets contain double the amount of insulin inside. And we demonstrated that this was a direct effect of bisphenol A in the beta cells. So if now we go and we extract the islets, we have ex vivo islets cultured during 48 hours in the presence of increasing concentration of estradiol. We measure here insulin content. So what we see is an increase in the insulin content in an inverted U dose-dependent manner, which doesn't scare anyone working with estradiol. Okay, so 100 nanomolar has no effect. 1 nanomolar, 100 picomolar produce the increase in insulin content, and this was due to an increase in the messenger RNA that we demonstrate was the transcription of insulin. So estradiol is increasing transcription of insulin and is producing insulin biosynthesis and this nice inverted U dose response. What is the situation with BPA? So if we use, again, 48 hours culture islets from mice, in the presence of increasing concentration of bisphenol A, we find exactly the same inverted U dose response. And this is 1 nanomolar bisphenol A. We have practically double the amount of insulin with 1 nanomolar BPA. And this is 1 nanomolar estradiol, equally effective. And if we do now this experiment in vivo, we inject for 4 days 100 micrograms per kilo per day or 1 milligram per kilo per day, we see that we have the effect with the low dose. We increase insulin content. The high dose has no effect. So we have the inverted U dose response also with the in vivo situation. Okay, so what is the mechanism that is producing this regulation of pancreatic insulin content, which is the receptor? Yeah, beta cells, they express both ER alpha and ER beta receptors. And this is an important point. Where are they located? Okay, this is an islet of Langerhans. We label membranes with agglutinin, and we do an immuno for ER alpha and ER beta. And we can see here how in green ER alpha is located mostly in cytosol, also near the membrane, a little bit in the nucleus, and ER beta is mainly extranuclearly located. And they don't go to the nucleus when we apply estradiol, for instance. They remain extranuclear. And that's an important point, as we're going to see. So what is the effect of an ER alpha agonist, for instance, in the regulation of insulin content? So these are increasing concentration of the ER alpha agonist PPT, and we see 1 nanomolar PPT is already increasing, and we have the same inverted U dose response, only activating ER alpha. If we now use an ER beta agonist, we have no effect in this case. So this is pointing to ER alpha as the receptor involved. But to molecularly prove that and unequivocally prove that ER alpha is mediating the effect, we use islets from ER alpha knockout mice. So that we're doing here is measuring insulin content. This is in wild type islets, or islets from wild type animals. So vehicle and BPA 1 nanomolar. So it's increasing the amount of insulin in this islet during 48 hours again. And if we use islets from ER alpha knockout mice, we have no effect at all. It's completely abolished. So if we do it with ER beta knockout mice, we are still having the effect. So this experiment is unequivocally demonstrating that the action that we have on insulin content by bisphenol A 1 nanomolar is ER alpha mediated. And that was very curious because the insulin gene doesn't contain an ERE element in the DNA, so we need to go to other pathways to activate that. And we studied this. And indeed, we demonstrated that activating ER alpha And with PPT or with BPA, we had a rapid, in 15 minutes, activation of ERK1/2 MAP kinases. And those regulate insulin transcription. So if now we measure insulin content, and if we concentrate here, which is BPA, we can see the increase with 1 nanomolar, and how this increase is completely blocked if we use ERK1/2 inhibitors or SARC inhibitors. And the same with estradiol or the ER-alpha ligand PPT. So now we know the whole pathway. Estradiol and BPA at the same doses, 1 nanomolar, bind to ER-alpha extranuclearly, activates CSARC, activates ERK1/2, and regulate insulin gene transcription. And this, at least with estradiol, have been reproduced by the group of Fran Mauvais-Jarbès, now in Tulane University, and Ken Korak in NIEHS, and they have added NeuroD1 as the transcription factor involved here. And okay, this is what happens with insulin gene transcription, with insulin biosynthesis. Now we're going to move to the other effect or the other role of beta cell, which is the pancreatic insulin release. So already some years ago, in 2006, we inject, and this is an acute effect, bisphenol A, 10 micrograms per kilo. Sorry, this is not ml, this shouldn't be here. 10 micrograms per kilo only. So when we see that, after half an hour of the injection, we measure insulin in plasma. And this is the insulin we had with estradiol and with bisphenol A, exactly the same doses, 10 micrograms per kilo, very low dose. So a very rapid increase in plasma insulin that was correlated with a decrease in glucose. And again, we wanted to know if this was a direct effect in the beta cell. So some years later, we did experiments ex vivo, and this is, in this case, is insulin release with increasing concentration of BPA in mouse islets. So we see here, in the presence of waves of stimulatory glucose concentration, we have an increase of insulin already significant with 1 and 10 nanomolar bisphenol A. So it is a direct effect in the beta cell. And how that is working molecularly? Well, I tell you how the stimulus secretion coupling works in beta cell. When extracellular glucose increases, it enters in the cell through GLUT2 in the case of mice, is metabolized, increases ATP-ADP ratio, and that blocks the ATP-sensitive potassium channel at the membrane, which is responsible for the resting membrane potential of the cell. The membrane is depolarized, and this is a signal to open voltage-gated calcium channels. Calcium gets in, as we saw before, and is producing the release of insulin vesicles. And now what I'm going to show is the effect of BPA in this molecule here, the ATP-sensitive potassium channel. This is a very important molecule. It contains the sulfonylurea receptor. Is the target for oral anti-diabetic agents. Okay, and we can measure the activity of this channel using the patch clamp technique. So that we do here is see the opening of the channels. This is the closed state of the channel, and we are measuring the current through the channels. So the more lines, the higher the activity. And as we can see here, in 7 minutes, 1 nanomolar BPA closes the channel, and there is a decrease in the activity of the channel. This is exactly the same that glucose does, and also oral anti-diabetic agents, and also incretins is the same, is blocking the same target. And this is completely dependent on ER beta. If we use ER beta cells from ER beta knockout mice, we completely abolish the effect of bisphenol A. And if now we go to measure insulin release. This is insulin release, and this is with 3 millimolar glucose, low glucose, we have no effect. Stimulatory glucose concentration, 3 millimolar glucose, increase in insulin, and 8 millimolar, and 8 millimolar glucose plus 1 nanomolar BPA, we have a high increase in only 1 hour, twice the amount of insulin being released, and completely dependent on ER beta. It doesn't happen in islets from ER beta knockout mice. So that's they're actually demonstrating that ER beta is binding BPA and having a very rapid extranuclear action in pancreatic beta cells from mice. But now we want to know what is going on in humans. So now we're going to human beta cells, and these are not cell lines or anything like that. These are real primary beta cells that are from cadaveric donors, okay? So if we use these beta cells and we do patch clamp experiment, we can see how in 7 minutes, this is the decrease in the activity of the channel. It is even stronger than in mice. This is 1 nanomolar BPA in human cells compared with vehicle, the activity. It's actually the same as 8 millimolar glucose. And if now we go and we measure insulin release, this is, I want to insist, this is a very physiological preparation. Now we use whole islet of Langerhans from human cadaveric donors. These islet of Langerhans are going to be transplanted to patients, and they are working and perfectly functional, are going to work in a person that is going to receive this islet. So when we use them, we of course see that if we increase glucose, insulin is released, and now with 8 millimolar glucose, we add 1 nanomolar bisphenol A, and this is the effect in human islet of Langerhans, practically double the amount of insulin being released. So I think the situation that we have after many years of work in the rapid response, we know practically the whole pathway, is that estradiol and BPA at low doses, 1 nanomolar, are activating ER alpha, and ER alpha we know that somehow is regulating guanylate cyclase A at the plasma membrane, increases cyclic GMP, activating PKG that regulates the ATP sensitive potassium channel at the membrane, regulates calcium signaling, and what is important for us now, and I have demonstrated in mice and in human, is very rapidly releasing insulin when this insulin shouldn't be released. And this is the whole scenario. ER alpha and ER beta can work extranuclearly with low doses of BPA to produce the regulation of insulin gene transcription and the rapid regulation of insulin release. And to finalize, the most important, that if the people that have done the work with my colleague Paloma Alonso Magdalena, who is running the lab with me now and has done a great deal of work during this year with BPA. Sergi Soriano has done all the electrophysiology, and this work has been done in collaboration for the ER beta part with Jan Ake Gustafsson, now in the University of Houston, the human beta cells with Albert Salehi in Lund and Anna Novials in Barcelona, and the ER alpha with the lab of Serge Neff in the University of Geneva here in Switzerland. Thank you very much for your attention.